Okay, so we're still in where are we? Chapter 12, right? Yeah, all right. So we're still in chapter 12. We're still starting talking about cell division, and I just want to go back one more time and kind of refresh our minds on what all these terms mean because they get very confusing, and it's important that you get a good understanding of these terms and they become very familiar to you because when we move into genetics and we talk about how inheritance occurs, all of that is tied into it. So I'm going to be using a lot of the same terminology. So make yourself little flashcards or whatever it takes for you to remember what these things mean. Remember, one of the more, or if you recall, one of the more confusing sets of terms is what's the difference between a chromatid and a chromosome. I tell my students, well, if you're talking physically, right? If you're talking about the chemical makeup, nothing. They're both DNA. They're both nucleic acids. They're both adenine and guanine and cytosine, all that stuff we learned about back in what was it, chapter 5 or whatever chapter that was. So when it comes to that, they're not. And then later on, we're going to see things where right here you can see sister, and then later on there's going to be something called daughter. Um, Sister, daughter, chromatid, chromosome, it's less to do with what it is, okay, and more to do with what it's doing. All right, so example, when your cells are just doing their thing, when they're in the interphase part of mitosis, right, when they're doing cell things and making cellular products and just doing cell maintenance things every day, kind of run-of-the-mill things, they're all chromosomes. You got 46 in case of the humans. You got 46 of those things doing their thing. And don't worry about the dominant recessive part if you know about that. I'll get into that. But let's just say the chromosomes are all doing their thing. Okay, now it's time for the cell to divide. Remember, the cells aren't always dividing. You know, organisms aren't always reproducing, and cells aren't always reproducing. So when cells get ready to divide, what they do is they condense, like you see in the picture here. They wind up because it's easier to move them that way. And since they're going to divide, you got to throw in that karyokinesis, right? You got to karyo the nucleus. You got to do something to the nucleus. What does you got to do? Well, you got to take the chromosomes. You got to duplicate them. All right, now chromatid, chromosome. So there were those 46 chromosomes doing their thing, and all of a sudden the time came for everybody to start condensing and making a copy. Because guess what, guys? We're getting ready to make a copy of the cell. All right, now what happens at that point is all those chromosomes, which are still chromosomes, make a copy of themselves. And now the copies are referred to as chromatids. Okay, so you see what I mean about being more a matter of what it's doing? The chromosome and the chromatid physically is no different. It contains the same information. It's a copy. Sometimes mess ups happen. But anyway, that has something to do with evolution. I can talk about in bio too. But what's going on is you only have chromatids. Chromatids only exist when a chromosome has made a copy. Before reproduction was getting ready to happen, chromosomes were off by themselves doing their things. Now you had two copies of each, okay? You had a number 8 chromosome that you got from your dad, the paternal, and number 8 you got from your mom. So they're both the number 8s, but they were doing their own thing. All right, now as we get ready to reproduce, each of those chromosomes, each maternal and each paternal, makes a copy. And now we refer to those things as sister chromatids. Those things, those two things, those two red X's that you see in the picture. Notice down below what they're calling a chromosome. Oh, 10 times 69. Oh, I wonder if the cat's trying to tell us something. 690. Play the lotto. Play those numbers in the lotto. All right. That's only 3 numbers, though. I don't think that wins you anything. All right. So notice down below in the bracket. Oh, I just got to say it. I'm sorry. There's a book, a really cool book, if you want to read it. It's called Archie and Mehitable. Archie and Mehitable. I believe that's how it's spelled. You may have read it before, but it's a short little book, and it's a story. Oh gosh, what is it? A cockroach and something else. These insects that at night they jump on the keyboard. In the house they live in, the guy's an author, and they jump on the keyboard and they type things out. And it's philosophical stuff. It's an old book, but if you're into that kind of stuff, pick it up. Archie and Mehitable. We're looking up online. You'll see what I'm talking about. All right, now down below in the brackets, back to science. Down below in the bracket, notice they're calling this pair, this thing, they put a bracket around it, this thing, this collection of two sister chromatids, they're calling that a metaphase chromosome. Remember in class, I also told you that chromosome refers to a unique set of information, and sister chromatids aren't unique, with those exceptions that I mentioned, right? So does that make sense now? So like in the case of humans, when cells are in interphase, there's 46 chromosomes, right? 46 chromosomes. How many chromatids are there as the cell gets ready to divide? So 46 chromosomes is going to become how many chromatids? Wouldn't that have been funny if that's would have been what your cat typed? Wow. I think Archie's knows why. Right? Come on, guys. Is the internet slow this morning? What we got going on here? 46 chromosomes. We're going into mitosis. This is a human then. How many chromatids are we going to end up with? Yeah, 92 chromatids. There you go. That's right, Jessica. 92. Now, why do you say 92 question mark? Just kind of let me know, what was the confusion? Is the terms, because that'll help me understand what I need to clarify for y'all. How about the rest of you? Was it just not answering or not understanding the question? Because you might see that again. You might see something like that again, because that's important in understanding. Yeah, I get it. I get it. Try to come up with a way that helps you remember them. Let's see. How could we do this? Oh, gosh. Well, yeah. I was going to say no, because you can call it a chromatid. My goodness, I can't think of something right now. My brain has hit a lock. But try to see if you can find some way to do an associative learning. You remember I told you about that in class where you associate something. Sometimes it can be something silly. Like when I was in my biology classes, we had to learn all the eras that happened in geological history. And they had names like Holocene and Pleistocene and Cambrian and Devonian. But we would come up with mnemonic devices. Like that one I showed you with the metric system. So we came up with, huge pigs put mice on every planet. Pink Cadillacs drive straight on curves. Now, each of those letters at the beginning stands for one of those eras, so it helps prompt your memory. So that's what I'm trying to do here with chromatid and chromosome. Think of some way to think of chromatid as a pair, chromosome is alone. Chromosome is alone, chromatid is a pair. Kind of, I don't know, get a rhythm there? All right. Okay, so, all right. You understand what I mean by the fact, too, of it's not a matter of what they are, but what they're doing. You're only going to get chromatids when they're duplicated. Okay, sister and daughter is very similar, too. It doesn't mean as much about family relationship as far as who's a sister to a daughter and whatever the case may be, but it's really more about what's going on. So you're only going to have sisters when you have chromatids. In this case, we're trying to think about them as being a pair. So a pair of sister chromatids, okay? When those are going to separate in later terminology, those sisters are going to become daughters. It's not a sister-daughter thing. Daughter means that you're a copy in that context. Sister means you're a copy too, but if you want to think about it as sister means you're a copy when you're connected. When you're in the picture that you see here, notice they're not calling them daughter chromosomes. They're not calling them chromosomes at all. They're calling them chromosome. Just keep looking at this picture and processing what the terms mean, okay, as you're trying to get those separated in your mind. Okay, it's the cohesion proteins that are holding the sister chromatids together. They are on the other side of that narrow part, which is the centromere. Centromere is just the narrow part. Again, it's not a thing, it's a region. It's some chromosomes, when they form sister chromatids, when they copy themselves, the narrow part will actually be up near the top. So the X will be a little kind of it won't be in the middle kind of X. The kinetochore are the proteins that are, if you think about it, is the attachment site to the chromatids for these things, which as we learned are microtubules, part of the cytoskeleton, but what they do is they pull these things apart, right? Pull those things apart. And they pull those things apart because when you're pulling them apart to make copies of cells, remember, you got to do karyokinesis first, and then you got to do cytokinesis. So you got to divide the chromosome, then you divide the cell. So the first arrow is karyokinesis. These two arrows are cytokinesis, okay? Cells aren't always dividing. The only time they're dividing is when they're in this phase, but don't forget that they actually are beginning to, I guess we might be able to say, beginning to get ready to divide. Yeah, see, making copies of all those chromosomes, that starts back here in S. That's why it's called DNA synthesis. So while the cell is kind of doing its cellular things, interphase, it's getting ready to divide. If you kind of think about it, it's the way, well, I don't know, I guess it's the way animals lead their lives, right? You either engaged in reproduction down here for a short bit of time, like we talked about, and in between, you're either just finished engaging in that or getting ready to engage in that, all right? So you understand what I'm saying there. It's a dynamic process. That's why it's sometimes hard to explain the phases static, you know, like say, okay, this is G2 of interphase, and this is prophase. And that's why we get stages like one more slide, prometaphase. Where does prophase, you know, I think someone asked, are we going to have to identify it under a microscope? You wouldn't be able to. You would, with our microscopes and the resolution they get, they're good scopes, don't get me wrong, but the point is you wouldn't be able to tell the subtleties between prophase and metaphase to call it prometaphase. Remember I told you in class I learned something like 16 different phases in mitosis? Because you can break up, okay, phase one. This, you can lump them together or you can split them. So we said prophase was basically when the chromosomes are all condensed, and if it's an animal, then they make asters and centrioles, which are found in a centrosome. That's an area, again, that yellow region there would be the centrosome. The centrioles would be the two little gizmos, I said, look like aluminum cans. I tried to help you visually here by highlighting in red those things that are only found in animals. And then down below, I tried to make the connection, and I write in red, animals only. So that's, you know, if you're one of those folks that's visual when you are trying to recall this information on the exam, like if I were to ask in a question, do plant cells have asters? Maybe it'll help to have that visual going, oh, asters red. Red said only in animals, right? Okay, so there's the sister chromatids, but notice that there's a good way to kind of get it straight in your head, right? Two sister chromatids, but one chromosome. So like in the picture here, let's do the exercise. I know we did it in class, but let's do it one more time, okay? And if you knew the answer or know the answer, don't answer. Let's see if we can, but if you don't know the answer, make a good guess. I don't know how to. I'm not sure how that's going to work out. But anyway, count the chromosomes that are in the nucleus of the cell. How many chromosomes do we have? There you go, 6. What did you count, Marie? What specifically did you count? Because it's kind of confusing because there's 2s of things in there. So what did you do? Yeah, okay, counted the pairs. I said in class what I used to do when I got confusing, and it's essentially saying the same thing you are, but what I used to say is I would count the centromeres. So if my teacher said, how many chromosomes there, I'd find the X's, and I'd say, okay, there's 1 there, there's 2 there, 6. 6, okay? Now, how many chromatids are there? Now, you could do this 2 ways, count them or mathematically, if you knew what the relationship with the chromatid and the chromosome was, right? 12 chromatids, right? 2 chromatids per pair of chromosomes. There you go. Good. All right. We'll move through here. Prometaphase. Metaphase, easy to figure out, easy to find out. One of the stages that I would expect you to know under a microscope because it's really easy to understand. It's really easy to recognize. They're all in the middle. They're in the middle, but don't expect them to be lined up like soldiers in the middle. They might be lined up more in a group because they're not all in the same plane. They're in a three-dimensional plane. So this is and I would not even, I can't believe I'm attempting to draw something three-dimensionally, but you get the idea. It's a cube. I'm not an engineer. Then those spindle fibers, or those mitotic spindles, those kinetochore microtubules, and even those polar microtubules. Remember, there's two kinds. They're calling them the overlapping, right, non-kinetochore microtubules, and these are the kinetochore. I also call these the polar because they go from pole to pole. Not as one, but they're not holding any of those chromatids. Now, the kinetochore microtubules down here, they divide the chromatids, right? They pull them apart. And the polar ones, they push the cell apart, right? They elongate the cell. I'm going to show you a picture of that. I'm working between two monitors here, and I get lost where my mouse is sometimes. All right, so there's metaphase in a plant cell. There they are. Now, there's the daughter, a copy, but not an attached copy. And notice we don't say daughter chromatids or sister chromosomes. We say sister chromatids and daughter chromosomes. So just write that down. Figure out some way to stick that in your brain, just like you memorized your phone number, which could be repetitious. You know, just saying it over and over. Sister chromatids, daughter chromosomes, sister chromatids. That'll work too. This is the Pac-Man thing. This is how those chromatids are pulled apart. The microtubules don't really pull, right? They just get shorter. So what's happening here is, as this motor protein is chopping off pieces of that kinetochore, which is tubulin, which is, go back to that chapter. That's what your microtubules are made of. This kinetochore, I love how the author keeps it in the same color. The kinetochore here, which is attached to the chromatid, is being pulled along. Kind of cool, right? All right, so you get early, middle, and late anaphase, and then, like I said, here's the polar. These are the overlapping microtubules, the polar ones. See, they go from pole to pole, and they just pull apart. So the cell is going to move the chromosomes apart, and then the cell is going to actually start pushing itself apart, elongating itself, because guess what? This is a dynamic process. So while karyokinesis is going on and those cells are pushing themselves apart, right, in anaphase, what's also happening, look, what's also happening is the cell is getting ready. This is an animal cell. Animals do cleavage furrowing, okay? This is what we mean by that. Those polar microtubules, when they're unoverlapping, they're pushing the cell apart. And then there are proteins who come in and start to cinch in the middle there. All right, so I'm going to get rid of my scribbling so you can see that. Okay, so this is an animal phase. So as those polar microtubules are unoverlapping and pushing the cell apart back in interphase, guess what they're doing? They're getting ready for telophase. And telophase, and it's kind of all even together happening at the same time. Back there in anaphase, as the polar nuclei are pushing the cell apart, we're also kind of starting the beginning of cytokinesis, because those proteins, Mother Nature don't waste no time, right? She's pushing that cell apart, and those proteins are forming like the string around the bag I was showing you, and they're pinching in like a sphincter. A sphincter. We all heard of a sphincter. We have lots of sphincters, not just where you might be giggling about right now. We have a sphincter. Sphincter. Sphincters are any muscles that when you do this, it tightens up like a purse string. You have a sphincter at the top of your stomach, so that when you lay down, it tightens up and stomach acid doesn't flow into your esophagus. When you get older, your muscle tone gets lost. You lose muscle tone when you get older. It's apart. It's called entropy. And your sphincters can sometimes get loose. That can be embarrassing, okay? Or when you lay down to go to sleep, your sphincter of your stomach opens up, it relaxes, it doesn't tighten like it should, and stomach acid will, that's called reflux, okay? Reflux. Okay, so all of these are happening at the same time. So the cells are being pushed apart, and telophase now is a karyokinesis thing. So what we mean by that is the nucleus is reforming. You can see the little purple dots, the nuclear envelope, of course. So is the nucleolus. The chromosomes, which they don't show in the picture here, they're going to start decondensing, okay? They're going to start unwinding. Remember they condensed? Remember I showed you in class, you can't read a condensed chromosome. It can't be all shoved together. I thought of another analogy. Like think about a file cabinet with a bunch of files. Like you work in an office and they're all jammed in there really tight. It's hard to pull them apart to see where they are. If they're kind of jammed in there loosely, you might even be able to pull them apart and read them. That's another analogy for condensation, maybe. That's how I always remember stuff too. I made analogies. Can't do cleavage furrowing in an animal cell. Or I'm sorry, excuse me, can't do cell plate formation in an animal cell. You do cell plate formation in a plant cell. You got to have a plant cell plate, because what that cell plate is doing is forming little holes, right? Little vesicles. Where's that picture? Why am I trying to draw? Holy cow. There's an animal cell, cleavage furrowing, daughter cells. So you can be daughter chromosomes. Look, daughter cells, copies, not attached. Here we go, daughters. There's what I'm talking about, that contractile ring, that sphincter thingamabob. See, it's making a... All right, move on. Right, inside, no, I have a picture of that. Yeah, no, this is the picture. Inside those vesicles that are forming, they're forming cell wall material, right? So inside those little purple dots that you see there, oh, not purple, those are the nuclei, but inside those little brown dots, or inside the dots in this photograph, cellulose is being laid down. Okay, so eventually what's going to happen as those vesicles get together, as they grow together, they're going to combine their cellulose and form cell walls. See? You can see a cell plate. You can tell when a plant cell is in cytokinesis because under the microscope, wait, where's that picture? Right there. You can see it. Look at this picture too. Name another structure you can see that's involved in mitosis. Don't name nucleus. I can see the nucleus. Name another structure that you see there that's involved in mitosis. So like if I pointed to it on a lab practical, could you tell me what it is? I'll give you a point for the first one that comes up with that answer. Tell me something that's involved directly in mitosis. Oh, Amber, nice. Yes, I see spindles. I see them right there. Good one too, because they're going away. But yeah, those are the spindle fibers. Nice. All right. Again, animal cell does it this way. Plant cell does it that way. And again, these are whitefish. Any of y'all go on the web and find these somewhere? Just type in whitefish mitosis. You'll find many of them are going to be labeled. I'm going to use them in class when we do lab too. I'm going to put them up on the board, labeled, and I'm going to say, go find this. It's really hard for me, or it's really hard for you, for me to tell you, go into your microscopes, get a whitefish slide, and find anaphase, right? If you don't have any idea what it looks like. Now, you might be able to, like hopefully, especially based on what we did the other day in class, you know that's anaphase. But it really helps if I can go find something that looks like this, right? And then I point to it and you go find it. Okay, so these are good study guides, but the ones online will be labeled in many cases too. So find those, prepare yourself for that, because this is real easy, folks. This is not difficult. And I'm not going to play games with you, like finding stages in between. I'm not going to try to trick you. I'm going to find some, the stages I use on the exam are going to be obvious. Like if I pick metaphase, I might pick any one of those three. All right, any questions on that? We're going to move into new stuff next. Where am I in time? Oh, good. Perfect. Cytokinesis. Maybe later cytokinesis. You said, how do you know it's not two cells next to each other? Well, let me get rid of my scribbling. Compare these two. Pen back. Compare these two with, let's say, well, okay, these two. If I asked you on an exam, give me a couple reasons why the one in the lower right is obviously in telophase, or the pair in the lower right, while the pair in the lower left is not in telophase. What kind of reasons could you give me to indicate that these two up here, right, they're not in telophase? Why? One point. Ah, Amber, didn't I say we can't, you guys can't get more than one, right? Didn't I do that before the first time? I think I'm looking back at my notes. I believe it was. But I do appreciate the answer, Amber. But yeah, let's just go with you can do that once per session, just so we don't, because I worry sometimes that the answer comes through mostly who has a faster internet connection. Aura, yes, you both of you can't see the chromosomes, right? Look at that purple thing right there. Okay, neither Amber nor Aura. Give me Aura. I don't know why I said Aura. Aura, give me another reason why I know these two aren't in telophase in the upper left. Give me another reason. And it's got to be a reason that, you know, the difference, it's got to be a compare and contrast, a difference between the two. You couldn't see the nuclei, I couldn't see the chromosomes in the ones in the upper left, but there's another reason, right? Which ones? Which ones? Kirian, which ones are not attached? The bottom ones are not attached? Okay, look at what's going on in the bottom ones right here. See that? See that V that's forming? What is that? And you can almost see it happening right across the middle too. What is that? What's going on there? Yeah, right? Now, does that look like this? That line right there is actually that other cell. So that look like what's going on up there? Yeah, no, see? What you can do many times in questions like that is look at more than one criteria. If one is like, I'm not sure, remember other things. Say, oh, you know, well, the shape, but that is kind of a V. Is it? No nuclei. Can't see the chromosomes. Well, the nuclei are there, but you can't see. Those are two cells in interphase. Those are two cells side by side, okay? All right, new stuff. Ready? Cells aren't always dividing. Cells don't have brains. They have all the characteristics of life, but they don't have brains. And what I mean by that is back to that idea, what I've said before, that one of the most beautiful things to me in biology is the fact it's just this incredibly complex dance of chemistry and physics. But let's do the chemistry part. Here's what controls the cell cycle. Here's what decides when the cell is going to divide and when it's going to be an interphase, right? So here over here, that little arrow or circle on the right there, those are the phases we just went through. M, G1, S, and G2. Y'all see that? Now, here's what we're going to talk about. We're going to talk about proteins again. Oh my gosh, proteins. That's not a surprise because proteins do all kinds of things. Proteins are found in all kinds of places. Proteins perform all types of functions. Guess what kinds of proteins, or guess what kind of function these proteins do? Well, they're going to regulate something. They're going to regulate the cell cycle. They're going to regulate when the cell is going to divide and when it's not. So this is what I'm talking about, man, this chemistry, this chemical dance that drives a living organism. To me, that's just fascinating. So let me in on the secret here. Here's the way it works. The proteins that we're going to talk about, the regulatory proteins, have really cool names. They're called kinases and cyclins. Kinases and cyclins. Okay, I got a question for you. What kind of a protein is a kinase? I mean, it might be a regular. There's a plus one. Amba and Aura, you guys are exempt. But what kind of a protein? It's a regulatory protein, yes, but specifically what type of a regulatory protein? Based on just its name. Kirian? Colin? Kelsey, how about you? Well, apparently we've lost cyber contact with those folks. Anybody else? ASC? Yeah, you're thinking root words, right? What's the root there that you're concentrating on, Aura? There you go. Right? It's an enzyme, isn't it? It ends in ASE. It's an enzyme. I could ask you that on an exam. I could ask you on an exam, yeah. I could say, all right, there's the cell cycle. You have these proteins are involved. What type of a protein is kinase? And where did that question come from? Well, it came from the fact that it ends in ASE. I want you to remember that stuff. Okay, now, cyclins is another regulatory protein. It doesn't end in ASE. Presumably, it's not an enzyme. Although, remember, that's not always the case, and I wouldn't expect you to know that. But remember back in photosynthesis, we talked about the enzyme Rubisco. That doesn't end in ASE. It actually really does. That's the believe it or not, enzymes have nicknames. Scientists don't want to walk around all the time going, oh, I don't even remember what the name of, I think it's ribulose 1,3-bisphosphate or some incredible tongue-twisting garbage like that. So Rubisco's true name, if you will, ends in ASE. So cyclin might be an enzyme for all I know. Right. Now, here's what happens. There are certain kinds of kinases out there, just like there are certain types of regulatory proteins. And one type of kinase is in bold type right there. They're called cyclin-dependent kinases. So whatever this enzyme kinase is, and consider that a group of enzymes. There are many, many different kinds of kinases, okay? Whatever this particular type of kinases are, they are dependent. In other words, in order to work, they need something called cyclin. That's why they're cyclin dependent. We call these things CDKs, cyclin dependent kinases. All right, now, the reason why cyclin dependent kinases are so important is because what they do is they turn something on. It's a chemical thing again, right? See, as cyclin dependent kinases increase in your body, what they do is they form something called this, an MPF, a maturation promoting factor. Now, what's a factor? It's a molecule. Think about again as the word factors meaning not what it is so much as what it's doing. It's a factor. It's going to do something. All right. So what happens here is the cyclin and the CDK form this complex. Okay. And that complex together is the MPF. So if you want to do math, you can say that cyclin plus some type of cyclin-dependent kinase equals a maturation-promoting factor. Okay, now, if I haven't totally swamped your brain right there, this is the molecule that does the trick. A maturation-promoting factor does exactly what it says. What factor does it, or what does it factor into? It matures things. It promotes the maturation of things. Okay, now what we're talking about promoting the maturation of is just another way of saying we're making another cell. You know, think about it, the word maturation. When we talk about living organisms, we generally refer to maturation from a biological sense as you're old enough to reproduce, right? I'm not saying necessarily mentally, but physically, when you go through puberty, then you are mature. You are sexually mature. So what's happening? The cells aren't sexually maturing, no, but they are reproducing, and that's why the terms make sense here. All right, now the MPF, the reason why it's important is it allows the cell to pass through something we call a checkpoint, G2. That's what MPF. So, all right, look at the figure on the left. Look at the figure on the right first. Let's orient ourselves where we are. There's mitosis, G1, S, and G2. So in the green, the blue, and the darker green, we're all at interphase. So the cell's doing its normal thing. Here's that CDK thing. So this is a kinase. It's an enzyme, and it's a cyclic dependent. Now notice it has a little notch in it. And notice that this CDK is not going to be active. The reason why it's not active is because it's dependent, okay, on something. And it's a cyclin-dependent kinase. So it's all it is in that thing I just circled is the kinase. The cyclin-dependent part hasn't happened yet. But as your cells grow and mature and get older, so with time, another molecule begins to accumulate. It builds up in concentration. That's called cyclin. Now this becomes very similar to what we were talking about in enzymes when it went between competitive and non-competitive inhibition, and did the enzyme bind to a substrate or did it bind to the inhibitor? Now, does anybody remember what was the governing factor? Let's say of a competitive inhibitor enzyme. What was the governing factor that decided whether the enzyme got inhibited or not? I'll give you a point for that. What was the factor that decided whether the enzyme turned out substrate or nothing? Either the rest of y'all are snoozing, you're falling off the planet. So I tell you what, even if you've already answered the question, I'll give you another point. Yes, but I specifically said in the question, what was the factor? Did I say it in the question? Okay, let me modify the question. If I didn't, I thought I did. In terms of concentration, what was the factor? Yeah, yes, you're right. It's how well they fit. I can give you a point for that because I screwed up the question. But in terms of concentration, what decided whether the enzyme got inhibited or not? Reaction is the result, Kieran. That's right. But what I'm saying at the beginning, so an enzyme has an active site, and there are inhibitors out there, and there are substrates. How could you govern the speed of that reaction? Slow it down, speed it up, inhibit it. Changing what? Yeah, you're right. There you go. Absolutely. Yes, it's changing the concentration. So what you do is, if you want to make sure that this, maturation promoting factor, this, I almost grabbed the wrong pen. One of these days I'm going to do that. I'm going to take the ballpoint pen and draw on my screen. If you want to make sure, see, this is MPF. Notice MPF is made up of two things, cyclin plus CDK. Now, you're going to get a little bit of MPF over here. Well, actually, let me erase my box. I meant to say you're going to get a little bit of MPF over here. So you're going to get some binding of cyclin to CDK. But notice what's going on. Notice the arrow's getting thicker, right? Cyclin is accumulating. So as cyclin accumulates, your chances of getting this are going to go up, right? For the same reason is if you wanted to inhibit an enzyme, a competitive inhibited enzyme, add more of that competitive inhibitor. Sometimes I think students, what they do is they tend to sometimes make things more difficult than they are. Just think about it in terms of like sheer numbers, right? If the cyclin is accumulating, then there's a better chance that CDK and cyclin are going to bind. And when you get that, that is the key. Once you get MPF, MPF unlocks this G2 checkpoint that we're going to talk about. So this is what's going to control the cell getting ready to divide, right? The cell's not always going to divide because of the fact that it doesn't have enough cyclin. Now, the only thing the chart on the left shows you is the relative concentrations of the two. It says the same thing that I told you. The cyclin concentration goes up and then drops. See, right after division occurs, okay, you would think, what happens to all that MPF? Well, right after mitosis, another chemical signal starts breaking down the cyclin. And when the cyclin is broken down, it disconnects from the CDK, and we're back to the beginning again. Right? So the cyclin concentration in the purple in the chart on the left goes up and then drops, stays level for very low for a while, then it starts accumulating. The cyclin concentration, the MPF concentration, of course, increases as the cyclin concentration does. And that differs from cell to cell. There's a time. Sometimes it's minutes, sometimes it's days. Isn't that neat? I love that. It's kind of how knowing the toy you get at Christmas, how it works. You know, it's that tearing it apart to figure out how it works. I always love that kind of stuff. Let's go back to those checkpoints. Here's the G2 one. All right, there's the G2 one that we talked about. So the G2 one is not going to be unlocked for a variety of reasons. And we talked about one already, was the chemical part. You've got to have that cyclin. But let's jump up to G1 for a second, and then we'll run through all. There's three of them here, right? So as we go through mitosis, the way this is drawn is they're, making our steps along the way. It's like an assembly line. All right. Once we get just about done with the G1, the cell's growing, the cell's differentiating, we run into a checkpoint. And that checkpoint is governed for the most part by nutrition. And the reason why that is nutrition, when you eat, okay, you give your cells what we call growth factors. You give your cell the food to survive. That's what that means in a fancy way. Remember, a factor in this case is something that factors into what happens. How do we stay alive? Well, you have to factor in feeding. Okay, you see what I'm saying there? Sometimes the terminology gets a little weird there, but that's what I'm trying to make that connection here. So basically what the G1 says is if there's a nutritional problem, the animal is starving, the plant is starving, in other words, there's just not enough food, there's not enough sunlight, whatever the case may be, then the cell basically says, I'm not going to reproduce right now. Okay? I'm going to reproduce right now because I sense chemically that there aren't a lot of growth factors out there. So that's telling me that the environment does not have a lot of food. Okay, so that G1 checkpoint is going to control not only the fact that the cell's not going to divide, it's going to stop the cell from getting any bigger. And what will happen sometimes is the cell will actually go off into a what we call a G0. All right, now what a G0 is, cells aren't always constantly dividing. They're not constantly going through that cycle. Oftentimes cells will go through the cycle and then they'll stop for a while. They'll concentrate on doing cell stuff. And then if enough of those cells die off or injury or something like that occurs, and it's, you're not always making kids. You're not always producing offspring. So sometimes cells will enter this sort of G0 suspended animation state, suspended only in reproduction. They're still doing all the other stuff. Okay, so at the G1 point, what basically the cell is deciding is whether it can afford, right, another offspring. And if there's enough food, then it runs through that G1 checkpoint. Now, you start synthesizing DNA. Makes sense to do this before you start synthesizing DNA, too, doesn't it? Makes sense to check and make sure you got enough food for energy to do this complex, synthesizing DNA is an endergonic reaction. You're going to need food for that. Now in the G2, of course, we talked about the CDK and the maturation promoting factor. Another thing that's going to be done in the G2 is the cell is going to make sure that all of the chromosomes got replicated. Remember, they're going to be divided. They're going to be divided eventually. One sister chromatid will go one way and one will go the other way. But you're going to want to make sure that all copies are made. So if you got 46 chromosomes, it's like 2, check, 3, check, 5, check, 8, check. Make sure everybody's copied. Because if you got 13 and there's no answer, you're like, oh, wait a minute, what happened 13? Why didn't you make a copy? We're going to need that copy. You're also going to make sure that you got some, you got good DNA. Make sure the molecule hasn't got a bunch of mess ups. Remember, I was telling you got those like those train car things that go through these. It's kind of like it's a protein, and it moves down the DNA and it looks for these mistakes. Now your DNA can make mistakes faster than it can keep up, but that's a different story. Okay, so if you get through the G2 checkpoint, then you finally enter into mitosis. There's one checkpoint left, an absolutely crucial checkpoint. This is the M checkpoint. And the M checkpoint stands not for mitosis, but I'm not sure if it really does stand for this, but in our book, it stands for metaphase. Okay, there's a checkpoint that occurs in metaphase that makes sure that each of those chromosomes, each of those pairs of sister chromatids, has a spindle fiber attached to it. I want you to kind of stew on that for a second. Why is that such a crucial checkpoint? So what it's going to do, that checkpoint's going to go through each one of those pairs of chromatids. It's going to say, do you have a gonadal-core microtubule? Yes. Do you have one? Yes. I make backwards X's because I'm left-handed, by the way, so if you wonder. If it comes down to a pair, yes. Yes. Here's another pair. Oh, something happened to the one on the right. We ain't got no spindle fiber. What do you think that does? How do you think? Why is that so important? Why is that such a mess up? That's worth a point too. Why is not having that spindle fiber such a mess up? Or if it's not connected, it might be there, but it's just the connection is screwed up. What happens right after metaphase? Yeah, they can't be pulled apart. That's for sure they can't be pulled apart. Yeah, and if they can't be pulled apart, that means one cell is going to get double the number of chromosomes, and another cell is going to be missing a chromosome. You ever heard of this condition in human beings called, I'll type it out. Have you ever heard of that? There's another name for it. What is that? Down syndrome, correct, yeah. What the word literally means, trisomy 21, is a person with Down syndrome has 3 number 21 chromosomes in their cells. Tri and somi, right? You're only supposed to have 2 because you only had 2 parents. The way someone ends up with Down syndrome is, in many cases, because the M checkpoint doesn't catch the fact that one of those chromosomes is missing a spindle fiber, or doesn't catch the fact that the connection is screwed up. We'll talk more about this in genetics, but that's that, in the most part, is the basis of most of these of most folks who end up with this problem where you have the wrong number of chromosomes. Okay. Now, speaking of having the wrong number of things, the next couple of slides are the wrong number of cells. This all is connected together, because the way folks get cancer is this whole cycle that controls when cells are being produced goes out of whack. See, there's a natural tendency, and I was reading a really interesting paper last summer, believe it or not, about how living organisms went to become multicellular. A lot of living organisms are unicellular, but it was, how did we become multicellular? But cells come with these pre-packaged, I don't know, properties. There you go. And one of those is the fact that cells require something to stick to for division. In other words, a cell won't divide unless it's attached to something. Now, you think about what could it be attached to? Well, in many cases, it's attached to other cells. Okay, and it's attached to other cells side by side, though. What's it attached to vertically is some sort of a surface. Oftentimes, it's the protein collagen. Many times, those collagen fibers, they'll provide a protein surface, and so you can form cells on there. But they'll only form in a single layer. That's because they have anchorage dependence, but density dependent inhibition. So their density dependent inhibition means they are not going to form a lump. They're not going to tumor, right? They'll only grow in a single layer. If a gap, a wound occurs or some injury and a gap forms, then the cells are only going to divide to fill in the gap. And that's what we'd call normal mammalian cells. The debate about whether other organisms out there get cancer and which organisms do or don't is still going on. So, we're just talking about mammalian cells. What happens with cancerous cells is a transformation. And oftentimes, this is what happens is a DNA mess up. Something happens to the DNA. Something happens to one of the proteins that are constructed from the DNA. Remember, the DNA is the information. But whatever happens, cancerous cells are transformed. And they no longer suffer from that inhibition, that density-dependent inhibition. And they form themselves in blobs called tumors. That's where you hear about it when you talk about someone who has cancer. They have a tumor, right? And the first thing the doctor is going to want to do, or one of the first things they do, is they want a biopsy of the tumor. They want a little chunk of the tumor, and they take it out, and they're going to grow it up in the lab, and they're going to see what type of tumor it is. And there are basically 2 types of tumors. Now, I'm not let me give you this word first. I'm not an oncologist. An oncologist is someone who studies oncology, and that's it's the study of oncogenes. All of us have cancerous genes in our DNA. It's part of our DNA. It was useful way back when, okay? But those genes are off. Sometimes things in our lives, smoking, too close to radiation, chemicals, exposure to the sun, will turn on those oncogenes. So an oncologist is someone who studies how those genes get turned on. And when those genes get turned on, what they do is they form tumors. Now, they take a little bit of the tumor out, and they want to know if the tumor is benign or malignant. That is important, and here's why. Let me go to the next slide. Breast cancer is a very well-known cancer. For the most part, it's relatively survivable now. My mother died of breast cancer 20 years ago, 22 years ago. Back then, it wasn't quite as survivable. My cousin had it a few years ago, and she seems to be doing quite fine with it. On average, in the United States, a woman has about a 12.5% chance of getting breast cancer. Men get it too, though, much less, 1 in 1,000, much less, but it doesn't mean that if you're male and you feel a lump in your breast, don't feel, well, men don't get breast cancer. They do. Here's a breast cancer cell, right, in the upper right. And as I said, what those cells do, if you look down here, is they form a tumor. The glandular tissue is mammary tissue. It's the tissue that produces the milk, okay? Now, what cancer cells are, you think, well, cells reproducing, right? That's not a bad thing. Well, the problem with that is as that tumor gets bigger and bigger, it requires more resources. It requires more blood. So it starts robbing the blood vessels from the surrounding healthy cells. So it actually starts causing these cells to become dead, or the word is necrotic. So the tumor robbing the blood from the surrounding cells causes that necrotic tissue, that healthy cells, and that destroys organs, and that's what causes the problem with cancer. What we mean by benign and malignant is malignant tumors. Benign is benign basically means no harm. The tumor does cause harm, but malignant means the harm can spread. It can undergo a process called metastasis. The way metastatic tumors work is pieces of the tumor break off, and they oftentimes will move through the bloodstream and the lymphatic system. That's why a lot of women, when they have, or a lot of folks, if they have their breasts removed because of breast cancer, they'll remove the nearby lymph nodes. You have lymph nodes under your armpits. We'll oftentimes take those off. And what will happen is little chunks of that metastatic tumor will spread throughout the body and set up place someplace else. So you'll have breast cancer. They'll remove the breast, let's say, or they'll get rid of the tumor. But since it was malignant, it shows up later. Say in my mother's case, it showed up later in her bones, and that's what she finally died from. So if you're going to have a tumor, I guess, right, in a macabre sort of way, the best one would be a benign tumor, because, you know, of course, that's what early detection is about too. If it is a malignant tumor and you get it early enough, you get it out before it spreads. And that's also what the 5-year thing is. A lot of cancer survivors talk about if you're cancer-free for 5 years, they take out the tumor and it was a malignant one, and maybe they remove the lymph nodes. They don't know it's spread until it shows up again. Generally, my mother was a little over 5 years. My mother was almost about 10 years, actually. But generally, if you're cancer-free for 5 years, they believe, not a certainty, they believe they got all of the metastatic tumor out of there. That's where that comes from. Any questions on that? Because we're going to move on to the next chapter. Okay, if you want to know why cancer even exists, why we have these oncogenes, like I said, you can take my Bio 2 class. So there is a time in the past in which being able to do this might have been an advantage. And living things are incredible, but we're not perfect. So sometimes things are left over from the past. This is what I used to tell my kids when they asked me where kids came from. Actually, what I used to tell them was magic. Whenever they asked me a question I didn't feel like answering, they say, Dad, how does a light bulb work? I'd say, magic. Oh, they were fine. But then their mother got ticked at me for doing that, like she's doing here. But we are going to talk about sex here. We're not going to be graphic about it unless you're a hydra or a bacteria. You're not going to have, there's not many graphical representations. But we're going to talk about life cycles, and we're going to talk about life cycles as it pertains to this process here. Sounds a lot like mitosis, right? The last chapter was mitosis, but something called meiosis. The British sometimes say meiosis. Maybe they're British, I'm not sure. But the reason why there's a mitosis and a meiosis is back to mathematics again. If you remember in mitosis, what we have is and I'm going to type all this out because it's going to be easier. We have diploid cells making diploid cells, right? So mathematically, we have a cell that's 2n equals 2n, right? Is everybody Follow me on what I'm saying there with the diploid, 2N, 2N. So I'm focusing really just on the chromosome number. Okay, now we all know the birds and the bees, and we know the way it works is sperm plus egg is junior, right? And how many chromosomes, if junior's a human, how many chromosomes should junior have? If he's a normal junior, let's say, whatever normal means. Okay, you have 46. And junior's parents, you had a mom and a dad, how many chromosomes did they have? Right, right. Okay, so now go back and answer that question. How many chromosomes does junior have? Thank you, Aura. Absolutely. If we went back to the original part and we said, okay, so junior's mom, junior's dad both had 46, junior had to have 92 chromosomes. That does happen, by the way. That does happen. It's a deformity, a severe deformity. But no, we usually end up with 46, and that's because of what Aura said. This process of meiosis has two basic functions. That's one of them, dividing the number of chromosomes in half. These things called your gametes, sperm or egg, they're not diploid, okay? They're haploid. They're not 2N, they're N. So sperm or egg, they're not 46 chromosomes. They're 23 chromosomes. So what Aura said is perfect. They each, 23 each, that's right. Mom and dad, 23 from dad, 23 from mom. So that's a really important function of meiosis, isn't it? You know, when this whole sexual life cycle part occurred here, the idea of combining two cells to make an offspring, there was a problem. What do we do about the chromosomes? Well, okay. Life says, let's have them first and then put them together. Now, not all your cells are this way. Most of the cells in your body are diploid. But the cells you produce that are going to lead to your sex cells and your sex cells is another word for gametes, by the way. Sex cells equals gametes. They're going to have half the number of chromosomes, so junior ends up with the right amount, okay? Now, there's two ways to reproduce out there. Well, there's many ways to technically reproduce, but along our lines here, there's sexual, and sexual is very simple. Sexual is a sperm, makes its way to an egg. The sperm has N chromosomes, and just like I said, junior now has 2N. That's what's sexual. The definition of sexual reproduction is however you do it, internally, externally, in the water. I don't mean internally in the water, but like sea urchins do that. They just, they spawn their eggs and their sperm out in the water and they float together. Maybe they get together. But if fertilization occurs, sexual reproduction, if fertilization occurs, the sperm and the egg come together and they form an offspring. That's what sexual is. The reason why we have to emphasize that is there's also asexual reproduction. And in asexual reproduction, this is a hydra on the left. Anybody know what kind of organisms hydra are related to? Very common organism down in the Florida Keys, actually. If I told you they can sting you, would you know? A hydra, if you were small enough. I guess a hydra does sting you if you touch it, but you just don't know it. Yeah, here you go. Hydra are what we call, a word, I love this word, especially because the first letter is silent. Hydra are cnidarians. Cnidarians include jellyfish and sea anemones. Notice bud. Now, that's not the critter's name over here on the left. The bud is the asexual part. Hydra can asexually reproduce. So eventually, this will grow, and then it'll disconnect, and we'll have another hydra growing right next to it. Two bacteria, what we called it back in mitosis? Binary fission, remember? They split in half. Binary is the one becomes two, right? So bacteria divide the same way cells divide in the rest of us. Remember, bacteria are a different type of cell. They're a prokaryotic cell. Now, there are some eukaryotic single cell like protozoans. Everybody always forgets about the protozoans. They're eukaryotes, but they are single cell. There's some of them that can reproduce this way too. Plants can reproduce asexually. If they're using flowers or pine cones or any of these other things in some of the other types of plants, then they can reproduce sexually. But when a plant grows bigger, when it when it grows limbs, or sometimes underground runners, right? Like here's the plant, an underground runner, and then another one will pop up. That's asexual reproduction. Now, the thing about asexual reproduction is you're a clone. What does it mean if you're a clone? So bud and parent, we say on the left with the hydra, they're clones. What's that mean? Yeah, you're an exact copy. And while that may not strike you as a problem, if you take Bio 2, you'll find out it is a problem. Because if everybody's alike, then whatever affects everybody negatively will affect everybody. Of course, that can cause extinction of a species, okay? All right. Close your eyes if you're bothered by naked men and women, but down the lower left, there are two of them. Obviously, the female's on the left and the male is on the right. Hopefully he has testicles, not just a testis. Unless he is, but know the name if only one of your testicles descend. Well, if you're female, they don't. But if you're male, your testicles descend through your abdominal wall down into your scrotum while you're developing. But there's a name if only one of those. Excellent. Thank you, Amber. I'm trying to remember how to spell it. Yeah, you're cryptorchid. There are rumors that Hitler was cryptorchid. Okay, so we're going to assume this guy's not cryptorchid. We're going to assume the female's got 2 ovaries, but we're just going to focus our attention on one. Now, colors, important here. The part in the pink, that refers to cells that are diploid. In other words, the male and the female down there, their testes and their ovaries are made up of cells that are diploid. Okay, now, in the process of meiosis that I'm going to explain to you today, what we're going to do is we're going to take those diploid cells that are in the testis and in the ovary, and we're going to run them through a process very similar to what you saw in the last chapter. So similar that I got to caution you to be very careful that you don't confuse them together. So towards the end, I have a few side-by-side things, comparing things. But this process of meiosis is going to do just what I said. It's going to divide the number of chromosomes in half. All right? So it's going to take, in the case of the humans, 46 chromosomes, and it's going to turn it into gametes that are haploid, sperm or egg, that are each 23 chromosomes. And when the sperm and the egg go back together for fertilization, then we keep the chromosome number the same. And then, of course, back to the previous chapter, Junior grows bigger by mitosis until Junior is big enough, or Juniorette. Is that female Junior? A Juniorette is big enough to undergo meiosis herself. All right, everybody knows the birds and the bees. Nothing here that's probably too unfamiliar. I hope not. Maybe a little bit of terminology. Diploid, Di2. Haploid, half, N. Okay, and the reason why we use the numbers is because not all of us have 46. Not all of us animals, not all of us creatures have the same number of chromosomes. Not all of us creatures reproduce the same way either. Just kind of a little interesting side fact for a second. This is what we just talked about, right? You have a diploid zygote. That's the first thing everybody is. As soon as the egg and the sperm fertilize each other, you're called a zygote. Put it in red for you. New word. We're going to see it again, too, by the way. Zygote. That's what you are. That first cell. So you make this diploid zygote. It grows up to a mature mouse, a male, female. They make gametes. They fertilize, and so on and so forth. In plants and in some algae, basically what they have is they have 2 stages that are multicellular. That's the big difference. You see, our sperm and our egg are unicellular. You and me as adults, we're multicellular, but the sperm or egg we make are unicellular. But there are some plants out there that actually produce a multicellular haploid structure. So if we did that, it'd be like linking a bunch of egg or sperm together. Now, without getting into a lot of detail, just to sum it up, the reason why that's done is in these organisms, it basically becomes a gamete-producing factory. They're all about numbers. These kinds of organisms are all about making lots and lots and lots and lots of gametes. Okay? More about this in Bio 2 if you're interested in that. I don't want to spend too much time. But I want to show you that there's more than one way to reproduce. There's a variety of ways. Now, in fungus and protozoans, they have a process kind of similar to plants and algae. But if you'll notice, the zygote never gets any bigger. You see right here in the mouse, after a zygote, mitosis made it bigger. This is a moss. After the zygote, mitosis made it bigger. But notice in fungi and some protozoans, you don't get that. The zygote is what we call short-lived. In these kinds of organisms, as soon as the zygote forms, it turns around and undergoes meiosis. Now, some fungi might take a while for that to happen, but right? So there's no diploid zygote there. So you got diploid zygote, you got diploid zygote, but you also have a diploid gametophyte, or I'm sorry, not diploid, multicellular zygote, multicellular gametophyte, and then single-celled zygote and single-celled gametes. Okay? All right. Don't worry too much about this. It's that I want you to focus on. Just wanted to let you know there's different ways of doing things. Now, chromosomes are paired, okay? We've been talking about 23 pairs all along. So if we looked at what's called a karyotype, that's what this is. You guys are so lucky. When I was in genetics, what we did is we would get a piece of paper with a karyotype. When you take a picture of the chromosomes, these are actual chromosomes that have been color-coded. But when you take a picture in a cell, when they're going to mitosis, they're not necessarily all lined up very nicely for you like this, okay? But we would take a piece of paper, a photograph, and cut it out with scissors and then try to find the matches. And you match it up by size, see bigger ones and littler ones, and you match them up. We didn't have the benefit of color coding, just by banding patterns. Ugh, give me a headache doing this stuff. But anyway, now computer programs do this. So what they do is they match up these chromosomes. Now, there's a name for these chromosomes. The ones in the that are in the number one, or rectangle on number one, that's referring to a pair of homologous chromosomes. Number two is another pair of homologous chromosomes, as is number three. Number 8 is a pair of homologous chromosomes. Number 18 is a pair. Number 11 is a pair. They're a pair because one of them's paternal and one is maternal, right? So maybe this is the number 2 paternal, right? Now this is your chromosomes, but the paternal and maternal represents where you got them, right? Everybody clear on that? All right, let me get rid of my scribbling. All right, now, what's this whole autosomes and sex chromosomes? In the case of human beings, okay, just in human beings, it depends on how many chromosome numbers you have, but pairs 1 through 22 are called autosomes. And autosomes are chromosomes that take care of everyday non-reproductive kinds of things. Now, doesn't mean that some autosomes aren't involved, but typically chromosome pairs 1 through chromosome pair number 22 down here, they're taking care of regular metabolic functions, digesting food and taking care of cells that are tissue that's being destroyed and processing food and all this sort of stuff. Any organism, regardless of the number of chromosomes, the last pair are always called the sex chromosomes. Now, the sex chromosomes have genetic information that have to do about sexual characteristics, but kind of like the autosomes, they also have genetic information that really has nothing to do with sex. We'll talk about color blindness later on. Does anybody know someone who's colorblind? Anybody have a friend or a family member who's colorblind? Matulia's colorblind? Ara? Oh, Miller. Really? I'll be doggone. Does anybody know someone who, I'll be doggone, I didn't know that. Now, female colorblindness happens as well, doesn't it? Does anybody know some female colorblind? Miller, I'm sorry, I can't remember if Coach Miller is male or female. I know I'm trying to look on the website here. Yeah, okay, all right. I'm sorry. Anybody know someone who's female and colorblind? They do happen. They do happen. It's very rare. The reason why, okay, last pair of the sex chromosomes. Look down here in the lower right. Here's the sex chromosomes. Everybody knows the way it works, right? So the male of this, or the male of this child, The sex of this child is just like Coach Miller. Not because I couldn't tell Coach Miller was male or female by looking at him. I just couldn't get a picture of him in my head. I don't want you thinking, can't tell Coach Miller that come beat me up or something. All right, so that's a male child. If that was a female child, what would those sex chromosomes be? What would those sex chromosomes be if that was a female child? There you go. Right? That's why they're called the sex chromosomes. The reason why I threw the color blindness in, the trait for color blindness is actually not on an autosome, it's on the X chromosome. And we'll learn in genetics why it's rarely found in females, but more commonly found in males. Okay? All right, everybody understand a karyotype? Everybody understand that these are paired, right? Most common type of color blindness is red-green, but there are other types of genetic color blindness that can be inherited. Does that remind me of your question in lab? I can show you in the book, the book we use to detect color blindness. It talks about some of the other types of color blindness. Yeah, it's really kind of interesting. Red-green is the typical kind. And just so it's one of these misinterpretations that I hear so many times, well, I don't hear many times, I have heard sometimes, a red-green colorblind person is not blind to, they don't see, it's not that the apple is invisible. It just doesn't look red. It's grayish. And it's not that the lime is invisible. It just looks grayish, okay? Everybody understood that? You can, even if you didn't, you could say yes, right? Who can't see red and green? He can't see red. No, but when he sees red, what color does he see? That's what I mean. If he says, well, I don't know, it's kind of a muted, grayish, washed out color. And then there's even degrees of colorblind. You can be more severe colorblind. Yeah. Ask him, because the fact that he sees green as purple is either the way my wife says, go get me the taupe purse. And I'm like, what color's taupe? She's like, brown. I'm like, why don't you just call it brown? Or it's because maybe there's some other visual color recognition problem that he can have. Color blindness is not the only one that affects that. Okay, so if we all understand the pairings, then we're back to a slide like we started the lecture with today, where there's a bunch of terms again. Now, maternal and paternal, we already went through, right? Everything's fine. I want to tell you that in this fictitious cell, the diploid number of chromosomes is 6. So 2n is 6. So there are 6 total chromosomes. Let's convince ourselves that we can come up with the number 6. Down below, how many chromosomes do you see? How many chromosomes do you see? Or I guess I should actually just say, do you see 6? If someone's got a pen that they can draw on the screen, number them for me. If you have a pen, or you can use your finger if your screen is a touch screen. Beautiful. All right, wonderful, Carrie. Now, how many chromatids do you see there on the screen? You can just type that in if you want. I see 12. Anybody else see 12? Yep, you sure do. Now, let me get rid of Kirian's answer there. How many pairs of homologous chromosomes do you see? How many pairs of homologous chromosomes do you see? Does everybody else see 3? Okay, it's real easy to get these terms confused. Okay, if you can draw on the screen, take your pen and draw a line between another pair of homologous chromosomes. There you go. And everybody sees the last pair, right? How are they comparing them together? Well, in this case, real simply by size, okay? But really be by size and banding pattern. But just to get rid of the banding pattern variability, we're just going to make it by size. Okay, so everybody understand what it means when I talk about a pair of homologous chromosomes? And everybody understands what I mean when I say that. Okay, one more little math thing for you. Got 3 more minutes. Look at the one on the bottom that's already labeled, pair of homologous chromosomes. So one pair of homologous chromosomes has how many chromatids? And how many chromosomes? Right? Beautiful, 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 beautiful. All right, perfect. Okay, one more slide, and then I'm going to let you guys go for the day because this is a great slide to end on. This is an overview. We're going to get into more detail when we come back next week, but this is an overview of what's happening. So here we have, we're going to follow just one pair of homologous chromosomes. We're doing this with human beings, then we will be following all those pairs. We're going to make it very simple, though. Make it very simple. Okay, during the process of interphase, notice stages. Many cases are named exactly the same. During the process of interphase, each of those chromosomes is going to condense and it's going to make a copy. Nothing different there from mitosis. So when you get to the end of interphase, everything looks like what basically did mitosis. But now in mitosis, here's where the difference is. Those sister chromatids each would have separated, right? They would have split apart from one another. Here's what happens in meiosis. In meiosis, the first thing that happens is the number of chromosomes gets divided in half. So basically, and this is kind of oversimplifying it, but it'll help for this point, one cell separates and contains that chromosome, one cell separates and contains that chromosome. Basically, it's why an old name or a name that's sometimes used for meiosis 1 is called reductive division, because the number of chromosomes is divided in half. This is precisely where diploid chromosomes, or I'm sorry, diploid cells, thank you, pen, become haploid cells. This is where 2N turns N. You got it? Again, don't get confused by chromosomes, chromatids. That's why I've spent so much time on that. Down in meiosis 1, indeed, yes, you have 2 chromatids, but you got 1 chromosome. Now, meiosis 2 occurs after meiosis 1. There is no interphase 2. Rule of thumb, never forget this. If on the exam assignment question, if I use a number to talk about one of the stages, like if I say prophase, okay, or if I say prophase 2 or metaphase 1, if I put a number, I'm talking about meiosis. Never forget that. Number equals meiosis. The stages you learn without the W, the stages you learned in mitosis are going to be repeated again, but we're going to put a number on them. We never put a number on the ones in mitosis. Prophase, metaphase. We're going to start putting 1s and 2s. Not today, though. That's why there's a meiosis 1 and a meiosis 2. There is no interphase 2. Because at the end of meiosis 1, telophase 1 immediately goes right back into prophase 2. Now, the end result of meiosis 2, it's called mitotic division. Not because mitosis is happening. Well, it is kind of, but it looks very much like mitosis, doesn't it? If we started, I was getting ready to cover the screen. If we started below here and I was looking at 2 separate cells, you would say, yeah, that's what happened in mitosis, right? Each of the spindle fibers pulled the chromatids apart. So that's why it's called mitotic division. All right, now the end result, and I'm going to let you guys go, I know I'm over, is you have haploid cells. See, diploid to haploid. The other thing that happens, and this is the last thing I'll say, is each one of those haploid cells is different. They're genetically non-identical. We'll talk more about that later on, okay? It's why you and your brothers and sisters don't look alike, even though you have the same parents. Okay, any questions on that? I'm going to stop the recording.